with, together with, it was her, Su Yuan Pu, who is another postdoc in the lab, and me. And so it was funny to see her talking right before me about this very applied stuff, because it's very inspirational from my perspective. Okay, I'm going to be talking about a method to measure transcriptomes together with virus RNA from single cells in vitro and in vivo. And we named this technique virus-inclusive single-cell RNA-Seq, or visc-RNA-Seq, and turns out it's a terrible acronym because everybody mispronounces it in a different way. So this is the idea of the talk. There's going to be 4 parts. Part 1 is why this is relevant and how you achieve this single-cell type of detection of RNA of the host and the virus. Part 2 is an e-life paper that Elena, me, Su Yuan, and our bosses wrote together in e-life at the beginning of the year about kind of how this can be shown to work in a cell line. And then there's going to be a few slides about what I'm doing now, which is to do the same in PBMCs from dengue people or dengue-infected patients, and then kind of one slide about how maybe you guys may want to use this for your own labs. Okay, so why is it important to look at single cells? So there are several reasons, and here I list 2 that I think are quite relevant in this context. The first obvious one is that no matter how much cell lines you kind of study, viruses infect people, and that's why we care about them, and people have many different cell types inside their body. And there are cells that look like this, and so on, and they all do different things, and viruses could do very different things inside all of them. And so when you have bulk assays like bulk transcriptomics or proteomics or things like that, they're great, but you have always kind of a confounding factor of whether it's your cell populations doing things or whether your signal is coming from a specific cell type doing something different. Then there's a second reason why this single cell kind of high resolution is important, which is that you have heterogeneity even inside a cell line. For instance, if you have that different cells express a different amount of virus, in a bulk assay, this is called noise, and it's a problem. Whereas if you do single cell work, you can try to exploit this information to make an inference, for instance, about time-since infection. And so, although you did, for instance, a snapshot experiment, you can try to guess kind of the dynamics of the process. How do you get this protocol to work? Well, first of all, you have to sort single cells inside a 96-well or a 384-well plate. And then what you do is you do an RT-PCR using similar like you do for normal transcriptomics. And usually you have only one oligo that captures polyadenylated transcripts, so mRNA. And in this case, you add one or more oligos that capture your virus specifically. I studied dengue and Zika virus in particular, and they're not polyadenylated. So if you were to do a normal RNA-seq, you would totally miss them. Then downstream, you can do, if you want, some type of assays before sequencing that allow you to discover, how many cells are infected and how much, et cetera, so you don't spend all your money from your lab onto sequencing cells that don't matter, which is quite relevant. And then you throw everything that you like into a sequencer, and then you look at all the genes. And so the kind of proof of concept we've done on HUH7 cells infected with dengue virus and Zika virus, and the idea is very simple. You have uninfected controls and infected cells, and then you leave virus in for a while, and then you take separate type points, and then you sort out 400 single cells from each of these cultures, and you sequence around a quarter of them. Okay, and to make a long story short, the paper has like 5 figures, but there's no time for going all through that here. What you find is you find things like this. So here I show the dynamics of 2 genes, and on the x-axis you have how much virus you have in these cells. Every dot is a different cell from any of these 12 cultures. On the y-axis you have how much this gene or that gene is expressed. And I don't need to do much quantitation to convince you that this particular gene is overexpressed after a certain point. So in all the cells that have more than so much virus, the gene goes up quantitatively. And vice versa, actin beta, which I was using as a housekeeping for a while because I thought this is going to be super stable, turns out is the most anti-correlated gene in the whole transcriptome. So it's the worst housekeeping you can pick. Of course, it's the one I picked, but okay. So then Su Yuan, a postdoc in Shirit's lab, who is my co-first author of the paper, did the validation by knockdown and overexpression of some of these hits, and several of them are proviral and antiviral, et cetera, et cetera. And then more functional validation downstream is, of course, needed. But it's interesting that you can take a system in which you do no modification, no genetics, no nothing, and you just screen for it, and you find several hits that kind of confirm downstream. Okay, so moving to the slightly more current work, because there is no genetics, you can do it in vivo. And so we have several people from a cohort that Shirit and the collaborator is collecting in Colombia that are infected with dengue virus and Zika virus, etc. And we have some cases with dengue fever or severe dengue. We isolate PBMCs by FICL, and then we have a panel of antibodies to capture in more or less equal proportions many different types of immune cells. And then we repeat the protocol that we did before, and we end up with this matrix of the gene expression for all the genes plus the amount of virus inside the cells. And so here is really where the single cell shines, because of course the blood is a mess and you have all types of things in there. For instance, we capture hundreds of T cells, NK cells, NKT cells, B cells, monocytes, etc., etc. And so with one experiment, you can look at many different cell populations, and even more. Inside T cells, there is CD4, CD8, activated, naive, and the same is true for all populations. So you don't have to design 45 different panels and experiments and then validate all of them in replicates. You do one experiment in which you look at all this great diversity at once. And so what you can start looking at is whether you have signatures of, in this case, severe dengue in vivo in a specific cell type. And so here, to keep things simple, I just divided all the cells in the blood into 7 cell types, but you can divide them in 45 if you so wish. And you see signatures like this. For instance, IFITM1 is a kind of interferon-1 gene, and you see that it so the first column is always control patients, dengue fever, severe dengue. You see this gene is upregulated in severe dengue, but it's present also in several cell types in control people. So that could be a not very good marker because it's expressed, although at lower level, in everybody. But then you have genes like this one, IFIT3, which is essentially absent but in any cell type except monocytes, except in severe dengue, it's present everywhere. So now that you know this, you could make an assay maybe that looks for this gene in, I don't know, T cells or lymphocytes in general, and then maybe this is predictive for severe dengue. And vice versa, you have genes like CXCL10, which is completely absent from everybody and everywhere except in monocytes in severe dengue. So now this is all very early days and very exploratory, of course, but the power of single cell is, of course, that you can make these groupings and cell types without knowing beforehand. You just ask the computer to look at many different ways of splitting the data, and then you take the lead from there, and then you try to make an assay that kind of is a bit more feasible and less expensive than like 6,000 cells or something. So the other thing that we see in these patients is that we only see virus-infected cells in 2 severe dengue people, and only or almost only in B cells, and almost only in naive B cells, and those are very, very often infected, like more than half of them. So now you have these people that we expected should have dengue, maybe at low levels everywhere or things like that, and then instead they have very, very high prevalence of dengue infection in naive B cells. That makes no sense whatsoever, as much as I can understand the literature. But it's pretty clear. So this is the cells that are infected, and they have like these kind of naive markers, and they are IgHT, IgM, IgD, IgM double positive, etc. And so again, I'm not an immunologist and I'm hardly a virologist, but it's quite interesting that you can go into humans and then ask completely unbiased, oh, what do I see, what cells are infected, and what do they do? And you can screen so many, and so such a diverse repertoire at the same time. Okay, and then just to give you a sense of where this is moving, if you ever want to do any of this in your virus or in your cell type or in your lab, there are three things that kind of are quite easy to do and stand out. One is just do it on more viruses across the phylogeny. I have a PhD in evolutionary biology, so it's quite interesting for me to ask how many of these host factors change during viral evolution in time. And so we did dengue and Zika, and then the next question would be, let's do hepatitis C and yellow fever, et cetera, et cetera. And then the second one is apply to kind of different systems, for instance, primary cells and organoids. With Makeda, my collaborator in Shirit's lab, we are trying to look at neural crest cells and Zika now, but you can do it in many different things. And finally, because this requires no genetics, again, you can take a brain of an unborn human baby and look, what happens in all the different cell types of the brain, if you want. And the protocol is the same. It's exactly the same experiment. And with that, I'd like to conclude. So I told you about this method that allows you to get transcriptomes together with virus sequence from virus RNA from single cells, in vitro and in vivo, and how it works. A little bit about how we did the in vitro work, and you can read the paper. A little bit about that we are doing it in dengue-infected people now, and it works great, I think. And then a bit of kind of what future perspectives one could expect from this type of work. And finally, to thank the people that I worked together with to make this happen. So Yuan and I worked together for the longest time to make the in vitro work. And then Elena was most active at the beginning of the collaboration. Makeda is working with me now on the in vivo work, and all three are in Shiritena's lab at Stanford. And these two bosses kind of try to kick us around in order to get the stuff done. And this is funding. Thank you, and I'll take any questions.
Questions?
Karim. Payback time.
From your dengue single cell work from patients, did you learn anything new about, it's thought that dendritic cells make are the primary vectors of dengue in the cell? Does your data prove that wrong, or?
So Shirit tells me, look at dendritic cells, those are the ones, and they look, but I don't see much.
So maybe because they're all symptomatic, maybe in asymptomatic people this works. Four of our people are very, very sick, like very severe dengue cases, and two more have dengue fever confirmed like by qPCR, but they are not very sick. And all of them have confirmed qPCR in the plasma. And I mean, the assay is not exact, so it's not exactly clear how sensitive this assay is. Like I cannot tell you if there are 3 molecules of virus, we'll detect them, but if there are 2, we'll not detect it. Part of the reason is that there are all these different strains of dengue, and we don't know beforehand which one it's going to be, so you have to design, generic primers, etc., etc. But certainly, and this is, this is the first time this is done, etc. But the impression is we went into the study expecting the dendritic cells and maybe some macrophages, and we came out seeing half of the naive B cell compartment completely wiped out. So, I mean, yeah. Ahmad.
So did you find any new housekeeping genes that is not very liquid?
Sorry, whether we see.
Did you find any housekeeping genes that stay the same throughout the infected cells?
Oh yeah, no, there is housekeepings that do their job. Only actin beta is not a very good one. Turns out very late in infection in vitro, all this cytoskeleton stuff goes to pot. So tubulin and actin beta, actin gamma 1, etc., etc., etc. But GAPDH works. So, you should ask a cell biologist, I guess. Okay.
Okay. Let's thank all the speakers.
Thank you very much.